Hello, this is Dr. Plag, the Executive Director of Business Development at Enzychem Lifesciences. And it is my honor today to introduce our company. And thank you for your time and opportunity to give this IR presentation. Okay, so the Enzychem Lifesciences was established 1999, and it is listed as a co-stock in 2018, and there's a market capitalization reached to the $1.2 billion very recently, and we have 127 employees in South Korea and the US side, and we have 2 GMP facilities in Korea, and we have 3 R&D centers in Korea. And we are working for the new drug development. Okay, so our board of members are trying to very hard working to develop the new drug, global drug development, and it has been 21 years managed by CEO Ki-young Son and Vice President Hae-kyung Kim, and we do have other corporate leadership, such as the Dr. Do-yeon Jo, managing the US site, and Dr. Hee-soo Kim is in charge of clinical trial, and Dr. Chang Gi-hong is the inventor of the EC18, and Dr. John Choi is in charge of the clinical trial in the US site, and Dr. Plag, myself, is in charge of the business development. And also Dr. Hyun-soo Hyun is a medical director, and Dr. Q is in charge of CMC, and Dr. Sun-young Yoon is in charge of global R&D site. And we do also have many excellent key opinion leaders in the world, especially Dr. Jeff Crawford is the head chairman of the SAB members. He's in the expertise in the chemotherapy-induced neutropenia. And Dr. Steve Sonis is in charge of the Cryom, and Dr. Larry Kwok is supporting our oncology project. Dr. Michael Charlton is a key opinion leader in the NASH field, and Dr. Ronald Manning and David Gardner is in charge of the acute radiation syndrome. Dr. Jae-hwa Kim is investigating biological mechanism of action of EC18, and Dr. Cameron Wolf is in charge of clinical trials COVID-19 in US. And very recently, Dr. Woo-joo Kim joined our SAB members for his COVID-19 expertise. Okay, so our new drug candidate EC18 is called Plag, also generic name mosliciguat. This compound is very safe and orally available and lipid-based first-in-class small molecule. So this molecule has the capsule in the picture. It contains 500 milligram, and also this compound is very excellent drug safety, and based on the non-clinical and the phase 1-2 clinical trials. And this compound has a unique mechanism of action. It's called PERA, which has a pattern recognition receptor endocytic trapping accelerators. Okay, the mechanism of action is very unique, and you cannot find this similar mechanism of action in other existing drugs. So it is a little bit difficult, but this picture shows a little bit the easy understanding of the mechanism of action. So EC18 forms at the chylomicron in the number 1, and it binds to the specific GPCR called PLAGR in the cell membrane, and which upregulates CD14 or alpha-arrestin, and then it accelerates of the pattern recognition receptor into the cell, inside of a cell, to remove any pathogen or damp molecules. And this accelerating regulate the expression of a cytokine and chemokine, which also reduce the cytokine and the chemokine expression. Eventually, it prevents neutrophil recruitment and then terminate the immune response very early stage. Because of that, the early termination of the immune response cause the prevent the further inflammation, such as acute respiratory distress syndrome or acute lung injury. So the EC18 is the first class mechanism of action, returns to early and fast immune homeostasis. And we do have many the peer review papers published in the several different organs, such as liver, lung, and skin, and cancer, and blood vessels, and mucosa, and joint. And also we have more than 200 patents related to this global study. And also we are very actively presenting our research work in the international conference, such as the American Association of the Cancer Society and the European Society of the Medical Oncology, and also the therapeutic agent for the NASH, and also American Society of the Virology. So this is the showing that our EC18 pipeline, and working on the very different indications, such as CRIOM, COVID-19, CIN, ARS, and NASH, and immune checkpoint inhibitor combination. For the CRIOM, we have received a fast track designation by FDA, and we are almost completed the phase 2 clinical trials in US. And you are planning to apply for the breakthrough therapy designation soon. And the COVID-19, we almost completed the phase 2 clinical trials in Korea, and we are looking for some of the pipeline timeline data result very soon. And also CIN is also we almost finished the phase 2 clinical trial, and you are looking for the global partnership. And for the ARS program, we have received orphan drug designation by FDA, and you are conducting phase 2 clinical trial animal rule study. And for the NASH and immune checkpoint inhibitor combination therapy, these two markets are very big, and currently you are looking for a global partnership for the possible licensing out and working together. Okay, first of all, I'm gonna talk about the chemo radiation induced oral mucositis indication first. Okay, this oral mucositis is most common and debilitating complications for cancer, chemo, and radiation therapy. So as you can see in the picture, there are lots of the ulcers in the mouth. Because of that, many patients, cancer patients, suffer from the pain from this ulcer, and cannot talk, and cannot eat, and cannot drink. So the patient suffering a very poor quality of life. So because of that, many cancer patients even stop cancer treatment. So the 90% of the head and neck cancer patients actually received the chemotherapy and at the same time radiation therapy. And most of patients develop grade 3 severe oral mucositis, which is called SOM. So currently there's no available drug for the CRIOM disease, and we think that this market has a huge unmet medical need. So it is reported that pathogenesis of CRIOM is related to the DAMP molecule, especially HMGB1. This molecule is cause some inflammation and may cause more cell death and eventually ulceration. So if you remove the HMGB1 on the DAMP molecule, then the tissue got healed and recovered from the CRIOM disease. And EC18 molecule actually working on this mechanism called PERA, acceleration of the intracellular trafficking to remove HMGB1 and resolve this CRIOM disease. So let's take a look at the current competitor landscape of the CRIOM field. And the second column, GC4419 from the Galactic company, this molecule is under phase 3 clinical trial currently, and this one is administered by injection, IV, but it has to be a daily 1 hour infusion, 5 days for 7 days, 5 days per week for 7 weeks. And also another one, SGX 942 is from the Sologenics. This compound completed the phase 3 clinical trial last year, and they announced that this compound did not meet any primary endpoint. So this our molecule EC18 has a unique mechanism of action. It's called PERA, and it has to be orally administered every day. So EC18 has a core competence or pretty strong advantage over other competitors. So as a summary of the Korean project, we almost finished phase 2 clinical trial in US, and we are applying for the breakthrough therapy designation very soon. And this clinical trial is the randomized, double-blinded study. So you don't know the result yet, but based on the interim internal analysis, we think that this EC18 molecule have a very strong reduction, relative reduction SOM rate, such as 51%. This is a very higher than other reduction rate of the Galactic compound or Sologenics. So we are expecting very positive superior efficacy result very soon. And at the same time, we are looking for the expanding our Korean therapeutic area to other related areas, such as radiation-induced inflammation disease, such as dermatitis and fibrosis and enteritis, esophagitis and pneumonitis and proctitis. Okay, let's move on to the second pipeline, COVID-19. So this COVID-19, the program is very unique because EC18 has a very different mechanism of action. Unlike the other COVID-19 competitors. So EC18 has both mechanism of action, such as anti-inflammation and antivirus. Because of these two double mechanism of action, EC18 can be a very powerful COVID-19 therapy. As you can see that this EC18 actually shortened the duration of the time in the endosome-dependent pathway inside of the cell, and also minimize the virus duplication period at the same time. So eventually, EC18 reduce the chemokine, cytokine, and block the chemokine, cytokine storms, and eventually prevent the ARDS and the acute lung injury. Also, at the same time, this EC18 reduce the virus duplication time, and eventually reduce the viral the release of the progeny. So the phase 2 clinical trials is conducting in the America and Korea at the same time. Especially the Korean clinical trial is almost finished, and you are expecting the top-line data very soon. And in America, you are conducting, recruiting the patient very actively. So as a summary of the COVID-19 program, we expect the Korean study, phase 2 study very soon, and you are still ongoing phase 2 clinical trials in America. And we confirm that EC18 has both anti-inflammatory and antivirus effect, and this one is confirmed by in vivo and in vitro study. So the EC18 may be a very unique COVID-19 therapy and prevent cytokine storm, which are leading to the severe pneumonia and ARDS. So the EC18 can be a very powerful COVID-19 therapy and can be used together with the vaccines and other therapies. Okay, let's move on to the other program. It's an immune checkpoint inhibitor combination. So the immune checkpoint inhibitor is a very powerful, very attractive therapy for the cancer patient. Currently, about 43% of the patients receive the immune checkpoint inhibitor therapy. Even though this immune checkpoint inhibitor is very powerful and attractive. There are some limitations of some issues of this therapy, such as low response rate and some durable resistance and also immune-related toxicity. Because of that, many companies working for the immune checkpoint inhibitor combination therapy. As you can see that according to the Cancer Research Institute, last year there are 2,900 combination trials started last year with 253 different targets. So many scientists are reporting what is the reason for the low response rate and durable resistance for the immune checkpoint inhibitors. And one of them is that the tumor microenvironment. So the immunosuppressive activities of adenosine in tumor microenvironment is one of the main factors to cause some limitation of the immune checkpoint inhibitors. As you can see that the tumor microenvironment, you can see many ATP is released by cell death and many immune-related cells. And this ATP degraded into adenosine by the catabolism enzyme such as CD73, CD39. Because of that, many adenosine actually effect on the anti-immune effect on the tumor-related, tumor-associated immune cells such as the Treg, CD4, CD8, and NK cells like MDSC and the macrophage. So many are the companies working for how to block the adenosine signaling in the tumor microenvironment. There are several approaches. One of them using small molecule inhibitors and antibody to block the catabolism enzyme CD73 and CD39. Also, the antagonist of the receptor to block the adenosine-mediated signaling. So as I said, that the immune checkpoint inhibitor has some limitation, and one of the reasons is that the tumor microenvironment. So we have to convert the immune suppressive tumor microenvironment to immune-enhanced environment. How do you do that? So many big companies like AstraZeneca and BMS, Merck, and Roche, they are working on the inhibitors of the enzyme, catabolism enzyme, and also they are working on the antagonist of the receptor, adenosine receptors. But however, both the approaches have some issues because they still many ATP and adenosine is still available in the cell, and they can cause another anti-tumor, anti-immune effect of the T cells. However, EC18 is a fundamentally remove the adenosine. Adenosine is the damp molecule, and it can be released, can be removed by our mechanism of action is called para. So as a result, EC18 may be solve the limitation of the immune checkpoint inhibitors, and can be a good partner for the combination therapy. Okay, this is the anti-tumor effect of EC18 with the PD-L1 antibody, and we treat them EC18 with both two different cells. The top one is that the PD-L1 responsive cell line, which is the bladder cell, bladder carcinoma, and the bottom one is that the melanoma cell line, which is very less responsive to antibody PD-L1. So PD-L1 doesn't work at all in the melanoma cell. As you can see, that the tumor size is the same, but EC18 reduced the tumor size very dramatically, and because of that, the EC18 can remove the adenosine dramatically. That's the reason the EC18 can reduce the tumor size. And you can see them some synergistic effect with the combination therapy. And also, in the last year, we presented the synergistic effect of immune checkpoint inhibitor combination between EC18 and the PD-1 in the lung cancer last year in the European Society of the Medical Oncology. And also in the last year, American Association of the Cancer Research, we published the synergistic effect of the ICI combination between EC18 and the PD-L1 at this time PD-L1, in the bladder cancer. As you can see that there's a very strong synergistic effect by the combination of these two molecules. So as a summary of the ICI combination, EC18 fundamentally clears adenosine, which is one of the main cause of the immune suppressive tumor microenvironment. And because of that, the combination of EC18 and the antibody PD-L1 or PD-1 may produce better treatment outcomes compared to other combination therapy, such as the ATP catabolism inhibitors or adenosine-related antagonists. So EC18 may transform the immune suppressive tumor microenvironment into the enhanced tumor microenvironment by removal of the adenosine, and it can be a very strong effect of the anti-tumor immunity of T cells. Okay, finally, I want to share the new program, which is called Galactic Cosmic Radiation, also related to the acute radiation syndrome. Okay, very recently, Morgan Stanley projected the space economy. Space economy will grow $1 trillion. It's 1,200 trillion won. It's a very huge money, and many big companies such as the Tesla, SpaceX, and the Blue Origin from Amazon, and Facebook and Google is try to establish such a satellite for the space area. And among the space economy, space healthcare is maybe will occupy about 20% of this economy, which is still very big market. So what happens in the space environment? Why you should consider any healthcare in the space environment? So there are some scientific reports said that there are many reasons for that, and they reported that there are some 418 NASA and the Russian astronauts, and many of them also passed away. But the reason for death is that they have cancer and cardiovascular disease. So it is known that galactic cosmic radiation is the main cause of the cell death and the DNA damage and the cytokine inhibition, then can increase the risk of cancer and cardiovascular disease. And also our collaborator, Professor Cheryl Nicholson from Arizona State University, confirmed that the Salmonella, the bacteria, has altered 167 genes in the space and increased their toxicity 3 times. Also, another scientific report indicates that space flight promotes the formation of the biofilm of the Pseudomonas aeruginosa, which is one of the bad bacteria, and increase microbial toxicity in space environment. Also, it is reported that the reactivation of the herpes virus is increased in the space environment too. So because of this reason, in the space, so there are galactic cosmic radiation, and at the same time there's a microgravity. Both cases cause the human immune deficiency, which has very weakened the immune system. On the other hand, the bacteria and the virus is getting very stronger in space environment. And that's why we have to consider the healthcare issues, and you need to find some right treatment for the future space disease. And recently, the Enzychem Lifesciences established the Galactic Cosmic Radiation Medicine Research Institute. And then the vision of this institute is the integrated space health total solution. And we want to act as a role as a center for the research, collaboration, hub of space medicine among industry, academia, and hospital and research institute. And hopefully you can develop and commercialize of the world's first prophylactic and therapeutic agent for galactic cosmic radiation. You are focusing on the 3 main disease areas, such as oncology, cardio, and the central nervous system, and the cardiovascular and the musculoskeletal system, and the immune system. And the many ongoing galactic cosmic radiation project is is conducting in the U.S., especially on the Trish program and also NASA program. And very recently, we got awarded a grant from NASA with the collaboration of the Arizona State University team. And also, you are working on the Harvard team to apply for the another initiating effect on the maintaining immune homeostasis against the galactic cosmic radiation process exposure to the Trish program. You do have many research work from the acute radiation syndrome, which is one of the very serious disease area. And we have received the orphan drug designation by FDA, and you are working on the phase 2 clinical trial in the animal rule study. And this ARS market is about $4 billion, but GCR in the future is market of 20 times bigger than ARS market. The ongoing ARS project is in the conducting USA, especially the Department of Defense and NIH, and also in the Korea, the GCR MRI Institute and CRIP, and also the KIRAMS. So this is one slide show that how our initiative is working very well in the ARS program. And as you can see that the initiative treated in the post-irradiation after first day and the second day and the third day, still initiative improved the survival rate, even though after 3 days after post-irradiation, which is very strong survival rate increase. So we also presented our research result in the last year's Radiation Research Society, 2 presentation. So as a summary of the galactic cosmic radiation and acute radiation syndrome, we hope to develop the prophylactic and the therapeutic agent of the 2 radiation area, and especially the USA government is very interested in our molecule for their future therapy for the ARS and the GCR program. And we think that ARS market is getting bigger, and the GCR market is coming very soon, and is 20 times bigger than ARS market in the future. So as a summary of our company, Enzychem Lifesciences, especially focus on the global R&D. And number 1, Enzychem has a platform technology and a mechanism of action called Plag, which is very unique. And it is well suited for the anti-cancer, anti-inflammation, anti-pandemic. And second, as it is a small molecule, we retain our own GMP facilities for the mass production in the future. And the third, you are waiting for the top-line data very soon for the COVID-19 and Curium this year. And also the ICI combination therapy, we can solve their limitation, such as low response rate and short duration resistance, and then the initiating can be a best solution to resolve the immune checkpoint inhibitor therapy. And finally, Galactic Cosmic Radiation Medicine Research Institute, as a pioneer of the space medicine industry, we expand our research scope into the development of advanced material to provide the space health total solution in the future, and we can hopefully you can reside in the other space, such as Moon and even Mars, by taking our medicine. So I finished my IR presentation, and thank you for your time and attention. And if you have questions, please send them to the BioKorea Center. And you are very open for the comment and suggestion for the future collaboration. And I wait to hear from you. Thank you very much.